Hello, everyone. We are coming to the last session where we will go more to practical issue and some national initiatives. And our first speaker is Claudia Ruivenkamp from the Netherlands, where they have a very active task force group on IVDR and where they're already working together for a long time with the competent authority. So Claudia, thank you for accepting the invitation to present the work of the IVDR.
Hi everyone, and thanks for the invitation. My name is Claudia Ruivenkamp and I'm a member of the Dutch IVDR task force. I'm also a clinical laboratory geneticist in the NMC in Leiden, and that's the reason why I am the lucky one today to present on behalf of the Dutch IVDR task force. In 2017, when the IVDR came into force, there were many questions and just a few answers. Some scientific societies in the Netherlands, they realized that this new regulation may have major consequences for the use, availability and costs of the IVD tests. And they concluded that the impact for clinical laboratories is potentially enormous and that there is no insight into these consequences for clinical laboratories. To meet some of the potential challenges, the scientific societies of medical laboratory specialists in the Netherlands joined forces in a multidisciplinary task force, the National IVDR Task Force. And this has many advantages because many questions are the same for every scientific society or laboratory specialism. It brings together different perspectives between and within scientific societies, and you can provide a uniform national interpretation. And not unimportant, you can form a central point of contact around the IVDR, also for the competent authorities. The Dutch IVDR task force consists of mandated members of the scientific societies of laboratory specialties in the Netherlands. So not only members of the clinical chemistry are involved, but also members of the microbiology, pathology, pharmacy, medical immunology, and of course the clinical genetics is also involved. The aims of the task force were to raise awareness among decision makers, so the Ministry of Health, Welfare and Sport, Health and Youth Care Inspectorate, so those are the competent authorities in the Netherlands, management boards and health insurance companies. A second aim was to create sense of urgency among laboratory specialists, because at that time the majority of laboratory specialists didn't even know that there was a new regulation that may have consequences for the use of IVD tests in their laboratory. But the most important aim of the task force was to provide tools and to develop guidelines, so make a translation or interpretation of Article 5, Section 5 for laboratory professionals, which they can use in their laboratories to comply with the IVDR or to implement the IVDR. So how did and do we still work? We meet and discuss the IVDR. We communicate with or ask for input from professionals via professional associations and different networks. We participate in meetings which are organized by the Ministry of Health, our competent authority, and we participate in meetings organized by other stakeholders, like the Federation of Dutch Universities, National NAN Committee, and different patient organizations. Our first meeting was in January of 2019, and there we discussed the sense of urgency. So we developed a flyer to all laboratory specialists, which we published in September that year, and that flyer had the following take-home messages. That the laws state what to do, but not how to do it, that you don't expect that the IVD regulation will be postponed, don't expect that the Dutch government will not strictly follow European regulations, and that you are yourself responsible for compliance to the IVDR, and that we as a task force will develop tools and guidelines to help you with that. For the guideline for interpretation, we focused on the lab-developed tests, so the in-house IVDs, and the requirements for those tests are described in Article 5, Section 5 and Annex I of the regulation. During multiple meetings with the IVDR task force, the text of the IVDR was discussed until a consensus on the interpretation was reached and a draft document was developed. Then multiple stakeholders and interested parties, including our competent authority, were consulted to check whether they agreed on the interpretation and to provide feedback on the document. And this resulted in a first version of our guidance document, which was published in June 2020. And a second version of the guidance document was published in December of that year. And probably the end of this year, a third version will be published because of new developments. And here you can see a screenshot of our guidance document, which is also published in a paper where you can find the guidance document in the supplementary data. So what's also nice to say here is that the consultant stakeholders greatly valued the proactive initiative, which was taken by our task force, and they welcomed the input from the medical laboratory experts. So now I will show you some screenshots of the guidance document from our task force. So here for industrial scale. So the law states that the in-house IVDs may only be applied on a non-industrial scale. And it's the view of the task force that tests developed in-house, which are used for patient care, are not applied on the industrial scale. We also made a definition for a lab-developed test or an in-house IVD in our guidance document. So it's an in-house developed and produced test. Tests that are labeled research use only and that are used for diagnostics. And a CE IVD certified test in which adjustments are made. And I have to make a note here because in the draft version of the European guidance, there it states that the IVDR do not regulate any possible off-label use of devices by healthcare institutes. So as such, the use of research use only products by healthcare practitioners is not regulated by the IVDR, and also off-label use of CE marked devices by healthcare practitioners is not in the scope of the IVDR. So when this guidance document is published and this is still there and our interpretation is right, then the second and the third definitions of a in-house IVD are no longer valid. Which genetic test will then still fall under the definition? That are the PCR master mixes, so the design of your PCR primers, homemade MLPA kits, and also in-house developed software that's used on site by its medical staff. Then very briefly something about software, because one of the members of the IVDR task force recently published a paper on the IVDR and in-house software development in a clinical practice. In that paper, they link the general safety and performance requirement of Annex I to the standard EHC 62304, which is a standard for medical device software. And you can find a very nice cross-reference table in the supplementary data. What's also nice of this paper is they describe 3 use cases. And one of these cases is a bioinformatic pipeline, which is used for variant calling in sequencing data. Then something about Annex I. It contains several important general requirements that are set for the safety and performance of IVDs. It's huge, it's 12 pages and 3 chapters long. But the requirements largely overlap with the requirements in the ISO 15189 standard. And since most medical laboratories in the Netherlands have an ISO 15189 accreditation, the task force made a cross-reference table where they link the requirements of Annex I to the sections in the ISO 15189 standard. And this is a practical tool for laboratories to demonstrate that they comply with the requirements set out in Annex I. Then here a screenshot of the cross-reference table. We call it Appendix A in our guidance document. And on the left side, you see the general safety and performance requirements as they are in the regulation. And on the right side, you see the sections from the ISO 15189 standard. There is a color legend there, so green means that it's covered with the ISO 15189. Yellow means that it's advised to comply with this part, orange that there is a limited application, and gray is not applicable. And you can use this table for a gap analysis in your laboratory by putting an extra column and refer to your existing or your new documentation in your laboratory. Then under 5A, the law states that the devices are not transferred to another legal entity. Well, the transfer of results with or without clinical interpretation is essential for good quality and accessibility of the Dutch healthcare system. So there we said that the transfer of result and interpretation is allowed to another legal entity, and also the transfer of relevant protocols, work instructions, clinical validations, and descriptions in scientific publications is allowed. Only the transfer of the in-house IVD itself is not allowed. Then something on 5F. There the laws state that the health institution draws up a declaration which it shall make publicly available. And there we designed an example or a template which is supplied in Appendix B, and which you can use and make public on the website of your healthcare institution. And here is a screenshot of Appendix B with the example of a declaration. So here in part A, you put your institute or your laboratory and a contact person. Then enlarge, you see the declaration, and there it states that you comply with the general safety and performance requirements as depicted in Annex I. And enlarged here part B, where you can make a list of all your in-house IVDs. So where are we now? I think there are still some unknowns, also for the competent authorities. We think it's a good thing that the Dutch authorities want medical experts to be involved in setting up guidance for IVDR interpretation. And it's also a good thing that the IVDR task force is seen as an important partner by the authorities. And to all of you, I would say, stay informed by symposia, websites, and publications, and discuss, preferably within your scientific society, what's reasonable to do. And then I want to end here by showing you all the members of the Dutch IVDR task force. And again, a screenshot of the paper which we published, and where you can find our guidance document in the supplementary data. And if you cannot find it, you can email me and I can send it to you, because I'm also the secretary of the IVDR task force. Thank you all for your attention.
Thank you, Claudia. Given the time, we propose to go to the next speaker, and the next speaker will be Isabel Domrich. She is from Germany. She has practical experience, and in Germany there is also an active working group preparing some templates, and she will handle this also in her presentation. So thank you.
Hello and welcome to my presentation. I'm very happy to be part of this webinar, and I would like to thank the organizers for inviting me and also for giving me the possibility to participate with a pre-recorded session. The title of my talk is Implementing the IVDR in Diagnostic Laboratories. My name is Isabel Domrich. I started in 2020, in August 2020, in the hematology lab in Kiel. We are an academic laboratory for specialized hematological diagnostics with patient care, reference diagnostics, translational research. We are accredited according to ISO 15189 since 2011, and we have a predominant usage of in-house devices. I have a PhD in biology. I work with plant lipids, but I also have a regulatory background as I worked as a regulatory affairs manager for a global pharmaceutical company for some years. My main responsibilities now in the hematology lab in Kiel are the regulatory compliance with the IVDR. So I'm involved in performance evaluation and risk management, and I'm also coordinating the external quality testing, round testing for EuroMID. Furthermore, I'm in regular contact with experts in the field as I'm a member of the EHA, so the European Hematology Association IVD Task Force, and also the IVD Task Force from the Biomed Alliance. And I'm also active in the AWMF IVD Ad Hoc Commission, which is a national task force. I will come to that later. It is a fact that laboratories are using in-house devices. That this is true is on one hand shown by the questionnaire that was sent out from the Biomed Alliance last year. With this questionnaire, we wanted on one hand to get an insight about the preparedness of laboratories for the IVDR, but also to find out what kind of IVDs the laboratories are using. And this is what you can see here. So on average percentage of assays used by respondents, that's you see here that on average 52% of the assays used by the respondents are CE-IVDs, and 48% of the assays used by the respondents are not CE-IVDs. If I say not CE-IVDs, then I mean, of course, the in-house devices, LDTs, but also research use only products that are used in diagnostic laboratories sometimes, or CE-IVD products that are used with minor modifications or that are used off-label. A nearly similar picture is shown by the case study from the Löwen Group that was published in 2020. They have in total 922 different laboratory tests, and from those 922, 537 tests are not CE-IVD, or as shown here, they use the assays used by this university hospital. Around 40% are CE-IVDs and nearly 60% are not CE-IVDs. This raises the question, why do laboratories use not CE-IVDs? I guess many people would think that it has financial reasons, that it is cheaper to develop an in-house device than use a CE-IVD. And yes, this is part of the reason. So what you can see here is again from the paper from the Löwen Group, and they showed different reasons why they had to use a not CE-IVD. They also showed it here in the different diagnostic sections, but in sake of time, I will just stick to the summary here. So again, you see, we had that in total, they have 922 assays. From those, 537 are not CE-IVD. You can also see here. And here are listed the reasons why they had to choose a not CE-IVD. And as you can see here, for 4 of those 537 assays, the reason is that it's cheaper. Then there are some other reasons, but the most prominent reason is that there's just no CE-IVD test available. It is another fact that in-house devices need to fulfill Article 5.5 of the IVDR and therefore the general safety and performance requirements that are described in Annex I. And in the next slides, I would like to show you a practical approach that should answer what is applicable by when and where to start and which steps to go. To obtain regulatory compliance, you can follow those 3 actions that we have described also in our hemisphere paper from last year. The actions are appoint a dedicated team, make an assay inventory, and obtain regulatory compliance. To appoint a dedicated team and stay informed, it is quite clear that for complying with the IVDR, you need extra vacancies. It is good to have a main responsible person that has the oversight, but it should be clear that this can't be done by just one person and that you need support from within the lab, like from quality management experts or risk management and also validation experts. To stay informed, there are many good publications and video tutorials available. Here's a little preview of my last slide where I collected some useful references in resources for you. Action 2 is make an assay inventory. So you need to check all the assays that you have in use if there are CE IVD or in-house devices. For sure, it's important to understand this gray area because yes, you will have CE IVDs used strictly according to the manufacturer's instructions, and you probably or maybe have some in-house devices, but quite certainly you might have something in between like research use only kits or off-label use CE IVDs or CE IVDs with minor modifications. But if you're using an in-house device, then you need to check on the market if there's an equivalent CE IVD available. And if this is the case, you need to compare your in-house device with the CE IVD and check if the CE IVD also meets the patient need at an appropriate level of performance. And if this is the case, you need to switch to the CE IVD. You probably heard that the transition timelines have been extended. This is true for CE IVDs, as you can see here. This is depending on the different risk classes. So depending on the risk classes for legacy CE IVDs, the date of application has been shifted to 2025 till 2027. And this is also true for in-house devices. So the full date of application for the IVDR is still 2022. But as you can see here, some of the requirements that are listed here in Article 5.5 has been postponed. So some are applicable right now. Some has been postponed till 2024. And one has been postponed till 2028. And this is the Article 5.5D. And Article 5.5D is exactly what I just showed you, that you need to compare your in-house with possible equivalent CE IVDs on the market. And then if you can't justify the usage of the in-house device, you need to switch. And this has been postponed. The action 3 is obtain regulatory compliance. And you can follow those 4 steps to obtain regulatory compliance. First, to understand the IVDR requirements, then perform a gap analysis, develop processes to close those gaps, and document the IVDR compliance. IVDR compliance. I think that understanding the IVDR requirements is the hardest part because you have to remember that laboratories usually don't have regulatory departments, but luckily there is a lot of help out there. There are many good symposiums and webinars from different medical societies, for example. There are nice papers and tutorials available. Again, the list with the useful resources at the end of my talk. For me personally, the work in these different IVD task forces was very rewarding and efficient to get a good insight into the field. And so I would advise everyone to exchange and talk as much as possible with other internal and also external experts. Step 2 is perform the lab-specific gap analysis. To do that, you can use some documents that are freely available that we generated within the AWMF. The AWMF is a network of scientific medical societies in Germany. It combines 180 scientific member societies and 3 associated societies from all medical specialities. In 2019, the ad hoc commission in vitro diagnostic was formed, and the goal of within this ad hoc commission, various subgroups were formed with the goal to generate documentation to help diagnostic laboratories. And here you can see the documents that we generated. We have some templates, we have guidance documents, checklists. The documents here in bold, I would like to introduce a bit in more detail now. Like those 2 guidance documents, the IVDR compliant risk management and IVDR compliant performance evaluation. Those documents should really help you understand the requirements because they show what needs to be done in order to have an IVDR compliant risk management or an IVDR compliant performance evaluation. They also show you, they show you what you need to do and how you can do it. And they show you where you find more additional information if needed. So for example, for the risk management, we make some cross connections to the ISO 22367, which is not mandatory for in-house devices, but still a lot of useful information is in there. Similar for the performance evaluation part. So Annex 13 of the IVDR, performance evaluation, performance study, and post-market performance follow-up has a lot of good information. So this is really a good source for more details, although it's also not mandatory for in-house devices. The next document is the Annex I checklist. This is very important document, and it shows you on the first step what is applicable, which of the requirements from Annex I are applicable for in-house devices. You have to remember that the IVDR is written for CE IVDs, so not every requirement is also applicable for in-house devices. Then this checklist gives you also further information and interpretation help for the single requirements, and especially the second tab is useful if you're already complying with the ISO 15189. This is how this list looks like. So it's a big Excel table, and in this table, every single requirement from the Annex I is listed, so almost 200 single requirements. For every of those requirements, we checked in the ISO 15189 if there's a comparable requirement, and this is then here evaluated with yes, no, or partly. If we say here yes, then we give here also the chapter of the ISO norm, so that you can use your accreditation checklist and you have directly a good starting point to perform your gap analysis. You should keep in mind that the ISO 15189 is regulating the processes in medical laboratories, and the IVDR is regulating the product. So it's not 100% comparable, but still there is a lot of overlap, and it's a really good and helpful starting point. The next step is to develop processes. Once you find your gaps, you have to close those gaps. And of course, this is very individual and based on the processes that you already have implemented in your laboratory. But probably you will find some gaps in risk management and also in performance evaluation. From May 2020 on, when Article 55D is also applicable, you for sure need some processes how to check for CE IVD alternatives and how to justify the usage of in-house devices. Last but not least, you need to generate and update documentation to show that you are compliant with the IVDR requirements. For that, also some documents from the AWMF group can help you. So like those two templates, I think your documentation should show on one hand what is applicable. Remember, the IVDR is written for CE IVD, so not every requirement is applicable also for in-house devices. Optional, it could show what is already covered by an ISO 15189 accreditation. It should definitely show how you fulfill this every single IVDR requirement and where this is documented. You see here again Annex I checklist. This is because the checklist I just showed you that I use for the gap analysis, you can also use for the documentation, because here are some columns in between that were hidden before that are exactly there for the documentation purposes. So how to work with this Annex I comparison checklist? You can use this checklist to find out if an IVDR requirement is comparable to an ISO requirement. If this is the case, you can use your accreditation checklist and see how you have fulfilled this ISO requirement in your laboratory. You can open those documents and check against the IVDR requirement. And by that, you know if you need to update or generate a new process and also update or generate new documents to fulfill the IVDR requirement. So the lessons learned. You should work with high-level documents that can be used maybe as a reference for all your in-house devices. Adapt the processes like risk management and performance evaluation and the documents that you already have. So don't start completely new, use what is already there and adapt it. Although there are open questions like what is really equivalent, what are valid justifications for the usage of in-house devices, you should discuss with internal and external experts and do what seems reasonable. You should remember that you are the expert on your assays. So do what seems reasonable and document and justify your decisions. Also, there are, as mentioned before, there are many useful information out there and also useful publication out there, which leads me to my last slide, where I have collected some publications that are worth reading. Here are the links to official documents, and here are the links to some useful websites, for example, the AWMF website, where you can find those documents, also in English, that I just showed you. The ESLO website has very a good collection of useful resources and also video tutorials. On the website from the Biomed Alliance, you can find, for example, the questionnaire that I mentioned, and also the EHA website has a nice summary of the IVDR related information. And with that, I'd like to thank you for your attention. And if you have any questions or comments, please feel free and contact me. Have a nice day. Bye-bye.
Hello, everyone. Claudia, no, not Claudia. Isabel was not able to join live today, but we can send her questions if you have specific questions for her. Now we come to the last speaker, Oliver Idle, and he will explain a little bit more about software validation and the use of software and in-house developing, what we can expect and need to do. So thank you for joining this course. A lot of people are waiting for some answers about the bioinformatic pipelines.
Okay, interesting. Good to know. All right. Thanks for having me. And as far as I see, I'm the only person actually presenting live today, which I guess is interesting. So let me share the relevant parts of my screen. All right, so this is my presentation. Thanks so much for the invite. I think it's cool that you invited me, but as always, I kind of feel like a bit unqualified. So just to add a quick disclaimer in the start, like I'm not an IVD expert. That being said, I am like, I guess you could consider me like a medical device expert, bringing software to market for medical device companies, mainly startups. And given that there actually is a significant overlap, at least for software, between the IVDR and the MDR, I think there's at least something I can talk about today, which should be actually very useful. That being said, the past two talks were actually like very, very good with like a lot of good information. I'm not sure if I can kind of like follow up on that, but see how it goes. Talking about being unqualified, who am I? I'm actually a medical doctor. I got into software development, so I taught myself software development. And worked as a software developer, and later founded this company called Open Regulatory, which is the company I'm working in right now. We're actually 4 people, and we help startups enter the market. So what sort of startups are those typically? As I said, those are actually typically medical device startups. So if none of these companies kind of ring a bell for you, that's totally okay, because that just means you don't live in Berlin, and these companies are mostly in the Berlin startup bubble of healthcare companies. They are very interesting companies, and all of those are our clients, and we help them pass audits. Again, that being said, most of them were medical devices, but we also brought a few IVDs to market under the IVDD at the time. But again, there's quite a bit of an overlap. So let's get started. And let's start with the question, which all of you may already know, is like, what the hell is software as an IVD? So software as an IVD under the IVDR is pretty much the exact definition. Well, it literally is the same definition as an IVD under the IVDR, except that at the top part of the section, I just omitted all the other stuff, but it simply includes software. So if you go to the top section, you see it also includes software. And if it fulfills like one of these one of these intended users, then you pretty much have software as an IVD on your hands. So what would an example from practice, from our like practical consulting experience be? For example, it's a software which analyzes microscopic images, for example, of cancer samples, tissue samples from a tumor, and it would make some sort of prediction on like maybe treatment response of that tissue. So it would maybe tell you, okay, a certain chemotherapy might be like a good idea for this. So that's like a typical IVD, which we would be dealing with. I'm not sure if in the context of this whole conference, this is actually the typical situation, because I think there are like a lot of labs developing their own software here. So I'm not super sure if this applies, but still maybe there's like a huge overlap here. Now, compared to the other talks, which again, I think were very, very solid, my talk is, I think, very different in the sense that I try to be extremely concrete and not talk in a very abstract way, so that you guys can go home today and kind of say like, oh, I actually understood what the next step for me would be. Like I take this document and I fill it out and I'm like done. I'm kind of like a very, I guess, like practical person in that way, at the danger of omitting details. So step one is obviously found your company. This is what we tell all startups. Interestingly, some of them haven't gotten it done yet because, for example, they're university spin-offs and they're still part of the university. So before you can bring an IVD to market, and probably all of you already know this, you sort of need to be like a company bringing that IVD to market. You cannot bring it to market as, say, like as part of a university. At least it'll be like very difficult. So I'm not going to spend too much time talking about that here. Step two, document your software. So what does that mean? Typically, that means being compliant with the IEC 62304, which already was mentioned in one of the talks preceding mine. And what does that mean again? So the IEC 62304 is this like ancient standard which which basically describes software development as it was like 20 years ago, or more like 30 to 40 years ago. So when it was like dinosaurs on the planet and like humans sending the first probes to the moon, essentially. So it's like very outdated, unfortunately, and it has a lot of ideas which aren't really like very useful and very modern. So how does a very practical example of this look? One would be like documenting software requirements. So again, I try to make this very concrete and not very abstract. How would you document software requirements? It's something the 62304 requires, and it would literally just be a list of features which your software has. So in this example, you can actually see our software here has 5 requirements, which is kind of not very much. So typically you would have a few more, and you would kind of like describe each requirement would like describe the features. So you would, for example, say, hey, when the app is first being launched, it shows like an introductory screen. It exists in a certain language, or like a user can actually switch the language. It shouldn't like use all of the CPU, which is, I guess, a very technical term. Passwords are stored in an encrypted way, and so on and so on. So the 62304 has different requirements, and one of them would just be like, okay, dude, please define your software requirements. And then you fulfill the 62304 requirement by having this like ancient document in which you list the features of your software. And then you have like a few other things like, okay, is a software requirement a risk control measure? Yes, no. Does it have like related risks? Blah, blah, blah. So I won't go into details here, but that's just so you get a very specific idea of what sort of documentation you have to create. By the way, all these templates, which I'm actually showing today, we're publishing them for free. And actually, they're all for free on our website. So I'll get into that later. Step 3 is do a risk analysis. So I have the impression that a lot of people here actually have a lot of regulatory knowledge already. So maybe I don't have to explain what a risk analysis is. But I'll still like show you one example, which is like pertaining to software and how a typical risk analysis would be done for software. So a typical standard for risk analysis would be the ISO 49971. So that's 14971. And that requires you to define something called hazards, hazardous situations, and harms. And the example I'm showing here is those 3 things being defined for like a COVID diagnosis app. Again, I kind of like made this app up, and it is probably not an IVD, it's probably actually a medical device, but still it's kind of, I personally think this is a very interesting example because the hazard, for example, would be the app actually shows you a wrong diagnosis. So you would have this app which shows like, hey, you're COVID positive or COVID negative. So the app would show a wrong diagnosis with a probability of 0.1, so 10%. It would lead to a hazardous situation that the patient actually believes this diagnosis. And then with a certain with another probability, it would lead to the harm, which would be delayed treatment of COVID. So a patient believing they're actually healthy while they actually have COVID, and then the patient would stay at home and not get treated early enough, and that would be delayed treatment. So that would be a harm. And that's pretty much all the stuff you try to document for your risk analysis. Again, I'm simplifying a lot, but I personally like to simplify if that kind of means that I can give good examples and maybe people understand it a bit better. All right, next, you conduct user tests. So the IEC 60366 is the relevant standard here. So what you just need to prove is that your software is usable by users. You can probably omit this in certain cases, especially for some IVDs. Again, I'm not an IVD expert, but I would probably think that with an IVD, with an IVD software, you have pretty good chances that it may not even have a user interface. So if you have like some sort of, I don't know, software like counting cells in a microscopic image and just like giving you a number or so, well, there isn't really a user interface for that unless I can like interact with it and kind of like see the cells it counted. If I only receive a number like printed out by a printer, there's not much usability to test. So only do this if you actually have a user interface. How does it look? Again, it's extremely simple. This is how an example table could look like. So you just define, well, first off, you need some users. Typically, you need at least 5. You define for each user, you define a certain protocol of instructions and acceptance criteria. So for example, you say like, okay, please like launch the app and do like a COVID assessment or so. And the acceptance criteria is, well, the user actually launches the app and successfully does a COVID assessment. And then you would kind of like see, well, did the user succeed in using this app? Again, this is not about actually proving that what your app does is correct. Like if you would be a COVID screening app, it's not about proving how accurate it is, but it's more about proving that users can actually use it. So they push like the right buttons and they don't get confused and don't get stuck. Next and final step, which actually now is IVD specific, you need to do your performance evaluation. And probably most of you actually know this better than I do, because we do have a template on our website, and I can briefly kind of show that in the next slide. But I pretty much leave this up to the manufacturer and kind of hope that they have some sort of, I call it like paper publishing experience, because if you have some sort of, if you've ever published like a peer-reviewed paper or any sort of similar publication, I think doing a performance evaluation is actually not that hard, because again, the structure is very similar. You describe the state of the art in the literature, describe your materials and methods, how you want to do the evaluation, and finally do the evaluation, end up with results and some graphs probably, and discuss how good your performance was and how useful those numbers actually were. We also have a template for that on our website. And again, it's all of our templates are like very minimalistic, just like this presentation. So they probably cover what you need to be compliant, but you definitely don't get bonus points for writing like tens of pages in regulatory documentation, because our templates, again, they just cover the bare minimum. Now, what I personally really like to talk about is, I call this the reality check of all the things that we discussed just now. Do they actually make medical software safer and better? And again, I'm only coming to this from the perspective of a software developer and a physician. So a lot of you probably in the audience actually have another perspective, specifically the one of like lab technicians. But I'm looking at this from the software side. So how would we like see this in an old school manufacturing company? And that being said, I haven't worked in an old school manufacturing company so far. So you're creating some sort of manufacturing, some sort of product, and you notice, okay, the product quality is bad. So what do you do? Well, you define rigid processes and quality control. And this is a lot of what regulatory requirements actually require, like the MDR or the IVDR. They want you to introduce certain processes to ensure that you actually create products which have a certain quality. And then, well, after you've done that, probably your product quality improves and everyone is happy. At least that's how I imagine it in an old school manufacturing company. Like if you manufacture, I don't know, cars also. It might make sense that maybe people sign off on every manufacturing step and that it's actually done properly. Now, how does this apply to software? So in a software company, software developers work in an office and they ship crappy software. I guess that's the bad situation, which is our starting point. And then again, you apply the regulatory approach of defining rigid processes and quality control. Now, the interesting thing is does this have the same consequence like an old school manufacturing company, that everything just magically gets better? In my opinion, we simply don't know. We simply don't have any data right now on showing how regulatory intervention actually improves or decreases software quality. One thing which I can say, which actually happens, is that the software development becomes much slower. And in my opinion, slower software development is not equal to safer software development. And it has a lot of like second order effects. And the largest second order effect, which I personally see working with startup companies, is actually the best software developers quit and get a job at Google, because there it's actually fun to develop software again because it's not so slow. So again, I'm not criticizing the regulation directly, but I'm just saying, well, I mean, we just don't know very much how we are doing right now. We don't know what the effect on software is right now. And we still have a long way to go if we want to regulate healthcare software in a useful way, because in my opinion right now, we're just like blind people trying to regulate it somehow, but without really having any data on what our interventions are causing. Now, let's take another like sidestep and talk about the elephant in the room, which hasn't been talked about, at least in the two preceding sessions. So at least there's something new I can like mention in this row of talks. The notified body situation. So again, most of our clients are medical devices and not IVDs, but we have worked with a few IVD manufacturers, and there it was a very interesting situation, which I personally noticed. So if you need a notified body, you have these 7 notified bodies which are available right now. And yes, 7 is not a very huge number. And we had some clients who actually contacted all 7 of these and got rejected by all 7 of them. Not on the grounds that like they were an unsuitable company or risky product or so, but simply due to the fact that there was no capacity from a notified body. So that's the situation we're in right now, which I think is a completely crazy situation, because that means right now we can't bring any new IVD, at least software, to market, because all notified bodies right now reject new customers. If this is kind of wrong in our data, let me know, but that's currently what I'm seeing. And I think this is completely crazy, because that pretty much means there's zero innovation happening right now, new product-wise. Also, will we actually have more IVD software needing a notified body assessment? So we probably don't know, but the tendency is, and probably most of you also already know this, is that IVDs generally are being up-classified a lot from the IVD to the IVDR. So I actually found this very interesting study, which is referenced in the top image. So you can see under the IVDD, most of the devices actually ended up being self-certified. They call it self-certified here. It's more like self-declaration of conformity, 93%. And now under the IVDR, probably will have like 84% of devices actually needing a notified body. So classes B, C, and D. Only class A will not need a notified body, which is more or less 16%. And it's, in my opinion, it's probably very unlikely that we will see a whole lot of class A software. So if we kind of like take up these numbers, we have 84% of devices needing a notified body now, whereas it was 7% in the past. If you just do like a very simple calculation and assume the absolute number of devices stays the same, that's 12x the number of devices which will now need notified body approval, with only 7 notified bodies. So I think this is I don't know how to solve this situation, but I think it's a very extremely bad starting point. Now, finally, I have one more point. How can we make compliance less painful? This is kind of like what our company does, and I guess the personal hobby of me. And again, this isn't a whole lot of advertisement, but it's just resources for you to actually get this stuff done. I actually got to this when I was learning software development. So when I, as a physician, taught myself software development, there were a lot of cool things which enabled me to do that in my like dark student flat. Specifically, there were four really helpful things. There was publicly available information. There were example projects, tooling, and communities for questions and answers. And what I see right now in the regulatory world is we actually have none of this really. I mean, for example, what we're doing today in this seminar, it probably gets close to like a community for question and answer. But it's just like one day where you kind of like meet other people and talk to them. And it's not like you can ask a question on the internet anytime and get a great answer yet, like you would maybe get today. So how can we improve this? And we at Open Regulatory, we try to improve this with a specific, with very specific solutions. So on our website, which I'll post shortly, there are articles which are all free, which kind of walk you through these things in very simple English language. For example projects, we actually publish all our templates for free. So I'll show you those briefly. We have a community for question and answers, which actually already has 600 plus people from medical device manufacturers in it for free, helping each other, especially with like audit questions. And finally, we actually have our own QMS software, which also comes in a free edition, which is called Formwork. I call this the Bermuda Triangle of like effective learning. You can access our templates at openregulatory.com/templates. And again, completely no strings attached. You can, they are sorted by standards and you can download all of them for free. There's absolutely no strings attached. We have templates for the 4 main standards. So that's the 62304, software development, 4971, risk management, 62366, usability evaluation. And let's see, did I forget? Of course, 13485, the quality management standard. You can access our community at openregulatory.com/community. It's a Slack community. Slack is this like chat tool where you can like send messages and well, receive messages. Again, it's totally free. You can apply and just enter your email and you get an invite. Formwork, our quality management software, is actually paid. So we actually earn some money with that because it has like a pro version, but it also has a free version, which already gets you started. One other thing which I'd really like to share with you today is we did a project which we called the Great Notified Body Survey. So we actually gathered anonymous submissions on of companies, how their experience was with different notified bodies. And we actually publish all of these for free. So if you go to our website, openregulatory.com, then you have to navigate to the top section, like articles and questions. You can actually see the results of, I think it's more than 30 or 40 like reviews of notified bodies so far. So there are huge differences in, I guess, customer satisfaction, and you should definitely have a look at those. Again, like everything, you can see everything for free, no strings attached. It's a great resource just for checking out notified bodies. All right, that's it. I guess I'm done on time. This is my email address in case you want to get in touch. Oh yeah, by the way, we pay our salaries by actually doing consulting. I should have mentioned that. So we are kind of like a profitable company besides giving out all this free stuff. I do hope that kind of a few of you actually could like benefit from this presentation a bit, like by being maybe a bit more concrete and a bit more abstract. Definitely head over to our website and have a look. And besides that, thanks for listening and have a nice day.
Thank you very much, Oliver. I also sincerely thank the other 2 speakers this afternoon or in this session, Claudia and Isabel. We have very few questions in the Q&A box. Does this mean that people are getting saturated? I would guess it's because the 2 presentations or the 3 presentations were quite clear, mostly, and it's very generous for all the guidelines that will be shared in all the documents. So I think the 3 presentations were quite interesting. And at the same time, if there's no Q&A or questions anymore, it's because this group here is becoming so knowledgeable after these few hours of seminars that we're all there. I see a question, 2 questions on the software. Let me read one. It's difficult to imagine safety class A software. Do you know any example of this, Oliver?
Yes. So, I mean, it typically depends on whether you end up having patient harm even after all of your risk mitigation measures. At least that's how I remember it. I have dealt with like quite a few Class A software packages in the past. That being said, most of them are medical devices, but some of them are IVDs too. So yes, I have seen those. I think it's generally like quite possible that it exists. So I wouldn't kind of say like, yeah, Class A software in itself like is this like thing that doesn't exist at all. I think it's solely possible. Not sure how much that answer helped.
Well, I asked to the other speakers, Claudia, Anja, Els are still online. Would you like to comment on this, Els?
Yes, I would like to add something because Olivier is more for the MDR. So if we talk about classification of software, what was Olivier saying? It's about the classification of the ISO 62304 and not about the classification that we have in our IVDR. So you have a classification for software, and beside that, you have a classification of the IVDR, which is also used the letters A, B, C, and D. So, but it was safety A class R in the software classification is also lowest level of risk. So that's a good point.
Yeah,
I fully agree, Els. Yeah. I fully agree, Els. That's really, it can be confusing. So they're talking about the Class A of the 62304 and not of Class A of an IVDR. Because under the IVDR, this software, if it's a standalone software, it will not become a Class A under the IVDR. It will be in high risk going through a notified body, but it can be a Class A then for the 62304, for instance.
Well, there is another technical question, but I think it's interesting for all of us. Someone is referring to the 62304 classification for software, but then with the specific content that if the bioinformatic type pipeline, and mostly talking about variant calling and variant interpretation, is developed, it's always reviewed by someone in the laboratory who's like a laboratory geneticist. So the claim here by the person that has asked the question is, how can this software then do harm to the patient if someone kind of sits on it? Again, Anja or Oliver, this is yeah, go ahead. I want to hear your answers too. Anja?
Well, maybe I get this question quite often because, well, it's just a diagnostic test. And I just refer to an in vivo diagnostic test. My husband is a nuclear medicine specialist. He sees a lot of images. And those images are really tricky because if that image is not providing you proper information, the health professional will not make a proper diagnosis. And if you look at the ISO 14971, and now the technical report, it's clearly explaining that if your IVD, if your software gives the wrong output, the specialist can't make the proper diagnosis. And there's no difference between in vivo or in vitro. So you can't say, well, there's someone in between and they make the result, then you should not develop it. So it's, yeah.
Oliver, you want to comment on that?
So it's a really good point from Anja. Yeah, I would kind of like add on that, like, yes, in theory, you can be Class A if you have the setup which you mentioned. So for example, you have like an expert reviewing your result, and in theory, I don't know how that would be possible, and in theory, your expert would be able to detect like a bad result and override it. Like in this like very theoretical scenario, I do see you being Class A. But the interesting question now would be, does this software even make a whole lot of sense? Because software typically tries to like make the expert like faster or more accurate, right? And if you make them faster, you would be actually doing a task which they would typically do. So, and if you would make them more accurate, you would be like analyzing maybe data that they wouldn't be seeing. So in many cases, it's very, very hard to argue that an expert might spot an error and override it, because then you would implicitly also say that your software actually isn't very useful, right? Because like if I as a physician can like override all these stuff, like, well, why did I need the software in the first place? But I still think those cases generally would exist, even though they are just rare.
Thank you. I think this clarifies a lot, especially for the bioinformaticians in the room, but also for the others, I think. The last question, or the one question that is left here, it's mostly one of scope, I think, and I can either address it to Claudia, Anja, or else. What if a laboratory that is working under 17025 using its own LDTs, but then provides services to other medical labs. Whereby that laboratory that's providing these services is not a formal health institution. Maybe one of you should translate this into real situations. Anja, you may know such situations, or Claudia, where indeed a lab kind of offers something to another lab but is not in itself a medical institution. What's the situation then? Could I think about sequencing facilities who are not just delivering kits but delivering services? I think that's the issue.
Claudia, do you want to discuss this?
Claudia
or no? Then maybe I can say.
You can start.
It all depends. Is it an IVD? Are you becoming then an IVD or not? That's the main thing. If I look at, for instance, the EU reference lab, they do a lot of work for the IVD regulations, but itself it's not an IVD, or they have to have the 70025. So if you are not registered at the medical laboratory, then it all depends what you do. If you are a critical subcontractor of the lab, then it's not in one single health institution, then it's not, then the IVD, then it becomes an IVD for the medical lab itself, if they are going to outsource some part of the work into a 70025 lab. That's also how notified bodies look at if a manufacturer outsources production or design development in a critical to a critical subcontractor. It's not always required to have an ISO 13485. It's also possible to have a 9001 or a proper functioning QMS system. So it all depends how it goes, and then what your activities are.
I would think that this is really work done on that same patient sample or any derivative that's then sent, and that the reference labs are actually not treating patient samples. They're no. I would say they're controlling or referencing the test.
Yeah.
So it's more about I lost the word.
Part of the testing for your IVD, for your in-house development.
Going abroad to another institution, we had that question as well. Claudia?
I don't want to add anything. I think it's true. I think the responsibility is then with the lab who's outsourcing to the other company and get the results back.
Mm-hmm. And do they need to go for CE, or can they suffice by calling an LDT? Maybe else this is useful.
You need also take in account what is industrial scale and when it looks as commercial, because industrial scale is linked to commercial, and depending what they are doing or what. But the question was not very clear for us what the person would like to ask us. So, but there are different issues.
I think
it's also mentioned about in the single health institution.
Yeah,
so,
if I may try and understand the question or rephrase it, I think it's dual. It's, for instance, we have a diagnostic laboratory. Our sequencing is done in another laboratory, another private instance, and that sequence information comes back. Then what shall this sequencing facility do?
If
this is the external situation. Yes. The other one is when the sequencing facility is in our own institution.
When you have an external sequencing facility and the part that they are doing, it's under the definition of a medical device or an in vitro medical device, then they need to follow the regulation. But it's really depending. A calibration of an instrument can perfectly done under the 17025. That's also a service. It's a little bit what they are doing as service.
But I guess the file that they have to write about that sequencing shall be the same as the file that we have to write about that same sequence.
Yes, but if you use a bioinformatic pipeline, then it's a question, which bioinformatic pipeline is this? You need to use a CE mark bioinformatic pipeline to do diagnostic. If you don't use a CE mark, it's an in-house developed, but you do it for another legal identity, and then that's not allowed. So there are different definitions that you need to bring together, and you need to put it on a flow chart to see what is ongoing here and what part is the service.
But the other question is, can labs that don't treat the samples until the final report, can they have a LDTs if they're commercial? Is that the other way to defining it?
But you will pay there, eh?
Yes.
You will pay the lab, and then it's industrial scale that that lab is providing.
Exactly. So they have to be compliant, but can they have LDTs then, or shall they have CE mark?
No, if you do commercial activity, it's not an LDT. That's
okay, that's a clarification I think that was asked by in the question. Thank you. I don't see any other questions else.
and only the health institution that can go for Article 5.5 for hospital exemption. So we must be lucky that we are perhaps a hospital institution and that we can comply for this, but others cannot comply for this. Okay. But you need to verify the definition of health institution.
And that health institution has to be established in the union. So it should not be in the UK, that lab should not be in the UK, it should not be in the US. It should be really in and it's one single entity. And the devices are not transferred to any other legal entity. So when you have the example that you say one part of the test is executed at one part and the other one is the other, then you already have two single entities executing that same test or the same, well, it's part of the test. So you can't rely then on Article 5.5.
Exactly. That's a clarification, I think, that we needed. Thank you. If there's no more questions, Elsa, hand over first to you. There is also the...
I see that Jeroen is here. Jeroen Poels from the guidelines. Perhaps if there are questions related to the guidelines, Jeroen was able to join later. So perhaps Jeroen, if you have a camera, perhaps put it on so that we can ask some questions. If not, we can ask it like this.
Jeroen, I think I have a question that could instantly go to you. It's the very last one that was put in there. We have previous ones that would have applied for you as well, but your colleagues have been able to answer. There is an upcoming EU AI regulation on artificial intelligence. And how will this be connected to the entire IVDR? So as soon as AI is implied and the regulations for that, where are we related to IVDR? That's how I understand the question. It's not the easiest one to give you as a starter.
No, indeed. I'm personally not involved in the artificial intelligence regulation. The way we see it is that at this moment, it's not really clear how it's interlinked with the IVDR. But there's a few groups working on that in Europe to get clarity on it. And hopefully this will be more clear in the near future, but I cannot provide you a better answer at this moment.
Anja, you raised your hand.
Yeah, well, actually this morning, Olga, and I think she did a very good presentation, but she mentioned it at last. AI is implemented and it has an effect on our IVDR and on the software. And also the European Health and Safety Act or something, she mentioned something. There are two new acts coming over which are lying on top of MDR and IVDR, which is very good to realize that can have an impact. And we have to find out in the whole, in not only IVD, but also MDR, that it has an effect. Because yes, for software, every software application you will apply to at a notified body, they will look also at AI requirements and implementations.
Mm-hmm. Jeroen, a question that came earlier today and for which I think I would like your opinion as well. It's about the division, and others have already tried to answer, or certainly have answered. But where shall we start if we have a sample and then DNA extraction, different sections, software tools, interpretation? What is the entire thing one single LDT? Can we just exclude the extraction and say that's a different thing, and then samples go into the specific test? This pipeline of testing in the laboratory, can it be chopped into pieces such that parts becomes class A, others remain class C? You have a view on this, or any suggestions to make?
I would indeed look at it in the bits and pieces that make up this entire pipeline, and classify or qualify your device that you are using according to its intended purpose. If you do DNA extraction in the beginning, then you just look at that part first. And well, as you know, a sample itself is not an IVD, so you can transfer samples to other hospitals. But if you do the sequencing somewhere else, then look at the sequencing there. So I would indeed chop it up in different pieces.
The question has also been put then, for the reagent and so on, we understand, but what about the pipette handlers, the automated liquid handling and so on? They also have to be IVDR. Els has hinted to that, but when shall we kick out our old machines and buy new ones that are CE labeled? And what's the rationale, or what's the pragmatic solution to that?
Well, before you kick them out, I would start looking at whether there's, well, equivalent devices out there, and whether the technical characteristics of those devices are really better than the ones you are using right now. So that will tell you whether or not you should change your machinery. But you should also bear in mind that a lot of Class A devices or general devices for IVDs or labs are not intended to be put on the market as an IVD. For instance, you have PCR machines, many of them without a CE mark, and this is perfectly usable as well. It's just depending on how the manufacturer places his device on the market. If he really intends it to be an IVD, then yes, it needs to be CE marked. But otherwise, you still could use a PCR machine that is not CE marked.
Yeah, but what we will see is, for instance, the sequencers that are today not CE marked, exactly the same machines will then be CE marked and be sold. How many leeway do we have before we replace the ones that were bought like 2 years ago?
I think this is really judged on a case-by-case basis by the competent authority. It's hard to give a general answer to this. To be honest, if this was to be judged by our competent authority, I think we would be quite lenient on this, because we obviously do see the difficulties that this will pose. We might be more strict in other ways, but it's really to be solved on a case-by-case basis.
Yeah, that's the personal note I wanted to make. I think in discussions with you, we feel that there is a lot of pragmatism in there, and this is one of the nice pragmatic examples. Anja, you were first to raise your hand.
I have a question specific for Jeroen about the MDCG, but maybe also mentioning about those PCR cyclers and so on. It's listed in the as examples as Class A in the classification, but of course the focus of the competent authorities will be on the comparison of your in-house developed test with the highest risks, if there's a comparable product with the highest risk available. I think that's the pragmatic approach, at least what I understand also the Dutch authorities will focus on. It's the highest risk products first, and then later on those standard machines, they will not force you immediately if you see, oh, there's now a Class A one, I have to go in there. I think that's the one. But maybe I think the question about the in-house MDCG document, when will it be published? When will it be finalized? Do we have to wait for the MDCG in October before it gets endorsed, so to say?
Well, we did get all the comments back by now from the workshop that was held last month. Those need to be still looked at, to be honest. So we will have a meeting of the task force, I think, probably in July, beginning of July. And hopefully by then we can indeed finish the draft. So I would expect indeed by the MDCG in October to have the document, yes, indeed. There's always a possibility, if we want to have this document published sooner, to just let the MDCG vote by mail or by mail. There doesn't have to be the general meeting, but I think October is probably a good guess. And, well, just go ahead, Els. Yeah.
Yes, I had a question that comes quite often to me, and it's back to the equipment. If you develop now an in-house test and you need to buy different equipments, the in-house test has a classification of a D, high risk, or a classification of C. And you have equipment that you need to buy, and you have equipment that is available, the same equipment, like a PCR machine, that is available CE mark and not CE mark. What do we need to advise to these laboratories? It's now June 2022. We know already that in the future, we need to work with this equipment or not. Can we, based on a risk analysis, and say, yes, the step in this process, in our in-house test, is not riskful, that part, so we can use research use only thermocycler, or should you advise go for the other one? Because it will be always the case. I think they will be both on the market, because these kind of equipments will be used by research groups as by diagnostic groups. So.
Personally, I would look at the life cycle of your device that you are planning to buy. As you know, by 2028, you should.
A thermocycle can stay for many years.
I know. In that case, the answer is simple. Then I would say, okay, go for the CE marked one.
From now on, you mean better to start from now on for machines that will last more than 10 years? It's obvious, I think, and reasonable to go to CEs right now. Yes, I think so.
Maybe also to add, as your lab, you have to prove that you are in compliance with the Annex I GSPR. So you also have to verify and validate, of course, under your 15189, that the whole process is very controlled and executed. It's the same with the notified body reviews. When they look at the technical files, they will check, okay, we have a kit. They will check, okay, the software is there. And always there is a machine involved. Although it's a class A or a research use only, a class A is considered to be more or less more controlled. Otherwise, you as a manufacturer for your CE file have to prove evidence that your machine is functioning as it is. So they will ask more questions at a notified body if you use a research use only, a general equipment product compared to an apparatus which has been a class A. And they still will verify the effect of the machine together with your class C device. So be aware, that's what the notified bodies do. They will never keep the machine out. They will always, because it's always an interaction, and then the software as well. And most of the time I see it depends on your software system making the analysis, because that software system is using different kits. So that will be sometimes separately classified and have a separate CE mark file as well. So it's all in one combination. So if you have to go through that, I would suggest to go for a class A as well.
Thank you. I think the last question that popped up just a minute ago is essential, and I wonder why it hasn't been there before. It's the question: if we say that our kits that are in-house is better than the kits that are CE marked, do we then have to test each one of them to prove that ours is better? That's the question. And the question was asked by a bioinformatician. So his question is, shall we test all possible pipelines to prove that ours is performing better? I think that's a bright question at the end, right? Maybe Claudia, you've looked at the scope of the different laboratories, and you've also been involved in those guidelines. Maybe Claudia, you would answer first on how far shall we compare? Do we really have to test it first?
Yeah, you should test it, because otherwise you don't know if it's better or not. But you can, we think in our lab, if I speak for my lab, that we can think of a lot of arguments why our pipeline is better than a commercial pipeline. Because a commercial pipeline is for us a black box. We don't know what's happening there, because we put data in and we get an output back. And there we do our analysis on. And in our in-house pipeline, we know exactly what kind of parts we coupled as a pipeline. So we know exactly what's going on with our data. And we have the opportunity to put in extra quality controls, rebuild it with our bioinformatician. So we think we can think of a lot of arguments to use our in-house pipeline. But saying that, our lab is also now looking at the CE IVD commercial pipeline, or we know that the commercial pipeline is getting an CE IVD, and we're also testing that just in case to see. So yeah,
so that evidently puts a serious burden on laboratories if you have to buy all the individual kits for these couple of hundred tests which are currently.
Exactly. We are in the lucky situation that a company where we do our sequencing is buying this pipeline or has this pipeline, so we can test it. But if we had to buy it as our laboratory, we couldn't afford it. We couldn't do it. Then the company who is selling it should offer us the opportunity to test it for free.
That's first for free, then you're under a different position. And secondly, that you answered for the software. But for all the other wet work kits that are available and CE mark, do we have to test them? Else raised hands, and I think else will have the practical answer to that.
Yes, I would like to answer, but I think Jeroen can confirm. We have the UDAMED database, which will give a lot of information about the pipelines that are on the market CE labeled, about all the kits that we would like to test, but they need to give all the details, much more details than we have now in the future. And that's the reason I think that the amendments say, yes, you have time till 2028 to really say your kit is better or not. But I leave the word to Jeroen.
First, Jeroen, yes.
No, I can only confirm. You have the summary of safety and performance in UDAMED by 2028.
Yeah, but if I may then say from the lab perspective, it means that we have to go through hundreds of pages to figure out if the test is different. That's an awful job. Unless, as Elizabeth pointed it out this morning, we would join forces at the international level. Hey? We all have to use the same kits. We may join forces on this. Maybe this is an idea. But it's tremendous if we have to go for hundreds of tests through all these documents. Olivier, you have not been able to reply yet to your software comparison. Are you doing this effectively right now when you help customers? Testing?
So I didn't quite get the question. It was about like comparing regulations.
The question is if you want to prove that your pipeline, say, performs better, you have to test all the others to prove they are not so good. Are you doing this currently in practice compared to all the available ones, or you just go for the CE accreditation of yours, which is different?
Yes, so far we didn't have to do that, but again, that could also be because is this about like in-house developed devices? Because so far we only did like the CE mark devices where you just do like your performance evaluation and everything, and I don't think you need to like do this sort of comparison.
That's right. That's what happens. It's a burden where you do the LDT and you don't care when you go for your own CE. Then you don't care what's in the other files, right? Okay.
Right. No, in that case, simply no experience.
Okay. Else, I hand over to you. I see no other questions. I hand over to you for some practical conclusions, and then I'll take it. Go ahead.
First of all, I would like to thank all the speakers to make this available, to be flexible, to record the presentation, to come to the discussion. So, Art, thank you well. You will receive by email all the persons who joined the conference, the webinar. You will receive an evaluation form. It's not only for evaluating how good the presentations are, but there are additional questions because, as I was mentioning in the beginning, the European Society of Human Genetics would like to set up a working group where we can see where we can help our genetic laboratories in Europe and also the national working groups to bring them together and see how we can support each other and finally also support the patients. So, and there are some questions in this evaluation form. Please complete it, send it back as the instructions give. And that would be welcome to start up with this working group. And finally, I would like to thank Jerome and Oscar from the European Society of Human Genetics, which make it available, which did hold the practical organization. I only need to look for the speakers and contact you, and afterwards everything was done by Jerome and Oscar and all the technology behind it. So thank you very much, Jerome and Oscar. And of course, we would like to thank the board of the European Society that we were able to organize this for free for everyone, so that we can reach as much as possible people.
I'm happy to thank you all again for the same reasons. I think else first to congratulate you. I think you brought together speakers with knowledgeable speakers with sufficiently complementary skills. So this was really we've been immersed, not drowned, immersed in IVDR, I think, by the program and the speakers. Thank you. My thanks evidently also to Jerome and Oscar. This works smoothly. Gunnar Hugo was also one of the initiators of this seminar. So we really thank European Society for kind of giving that impetus to this initiative. And I wish also to thank the audience. We were there in big numbers, a little less numbers now, but I think that's the other duties more than tiredness. The thing is, we will be working hard within European Society / Eurogentest and create a task force on IVDR to closely follow up and also be more proactive than we've been as a society right now. All these initiatives will be announced through the European Society of Human Genetics newsletter. So if you are a member of European Society, you will receive that information. So whenever you see IVDR pop up, please read it as it would concern you. And if you're not a member, of course, you can become a member, and Jerome will be happy if you do, and we all will. And if you're not becoming a member at all, you can still consult these newsletters on the website of the European Society. So if you really want to keep up with what we're doing at the IVDR level in our society, please follow us at these places. I think this is all. We will certainly also welcome if people want to join our activities. So send a mail to Els or me if you say, well, I know all this and I want to be part of that European Society task force. You'll be very welcome to contact us. With this, I want to again thank you all. It was a great seminar. I wish to thank again everybody and have a nice evening. If at least in Belgium the sun is out there. I'm not sure if this is true in all of Europe, but yeah, thank you and congratulations to all. Bye-bye.
Thank you.